The afternoon session, we are having a lecture. This will be followed by interactive session after the lecture. So, without taking much time, we should be started. And at 4, we are having this feedback from you. Just like this.
Well, it's like the last effort in. Now it's well, it's the last session, last effort for me, but then it's bad for you. Exam tomorrow. Sorry, it's fun, but then it's coming work. It's work, no choice. So we're reaching another point. Remember, we were discussing already this point. The probiotics need to be alive. But then, if the metabolites can perform better, what's better than this? Because if you need to keep the probiotic alive, you need to be keeping the appropriate phenomena. So all these things is actually it's a trouble for industry, for dairy, for pharmaceutical. They need to keep this microorganism in the right number, in the right conditions. So it's kind of problem. So they actually they look for the short way to resolve this problem, and they were saying, okay, we can go for the spore-forming probiotics. Because spore-forming probiotics, you form the spores, you keep the spores, and you sell the spores, and then these spores can grow and they can perform their work. Well, but again, not all the applications can be done by the spores. And you already know that some metabolites are produced by non-spore probiotics. So, lactic acid bacteria, they're not forming spores and they have their benefits. We're having as well Akkermansia is not forming form and needs to be alive. We're having Saccharomyces cerevisiae, they don't form spores and need to be alive. So this is actually a point that spore forming was not really solving the problem. So some more people that actually did and observed something interesting point, that even if you use the dead cells, in fact, they can do the same job like a probiotics, because very often probiotics could be associated with the effect of some metabolites. Well, what is the definition of postbiotics? This is the preparations of inanimate microorganisms and all their components that offer a health benefit. Everyone agreed that the postbiotics are dead cells. Well, some say that can be entire dead cells, some say that some metabolites, but normally the general idea is that postbiotic are entire cells, everything together. Because if you have the metabolites, you have the metabolites, you have the effect of enzyme, you have the effect of bacteriocin, you have the effect of polysaccharides. If you can call them a metabolites. And if you apply the entire cells, you apply it actually as postbiotics. Um, some of the important points that also need to pay attention, probiotic needs to be safe, needs to be safe microorganisms, because this probiotic as a live organism will enter the cells and will be interfering with your host immune system. So it's very important that the probiotics need to be safe space, and they need to have appropriate studies on their safety. When it's question about the postbiotic, in fact, the safety of the microorganism is not such a big deal. Well, I do prefer the postbiotic to be derived of the safe microorganism because you don't know sometimes maybe, even if as a dead cells, you can maybe have some metabolites that could be with some negative effects. So in this point of view, the postbiotic safety is a little bit easier to deal with. And as well, we can think about the fact that we don't need to deal with the keeping this organism, because postbiotic, you need to keep it alive, having a short shelf life. Postbiotic is already dead cells, so you can keep it for a longer period. So you need to be aware that the metabolite is not going to be destroyed. But already kind of conservation process is much more easy. Another point, if you think about the postbiotic, postbiotics you can easily quantify because you quantify specific activity. You quantify enzymatic activity, you quantify antimicrobial activity, you quantify something that analytical chemistry already have enough methods to be quantified. And you have the probiotics. I'm sure the probiotic group produce these metabolites when it's inside of your body. Maybe not. You have in the plate a very good production of short-chain fatty acids, but when this probiotic go inside your body, maybe you will not produce the short-chain fatty acids. So it's important to be sure that you're going to have this production. In postbiotic, you already have produced everything. You quantify it, you know the quantity, you know the quality, and you can apply it. And it's making the entire process much more convenient and easy for application and for manipulation. So more or less, this is a paper that is, okay, I will say almost published because we had already the first revision. There was, editors was, reviewers were suggesting minor revision. So we're just waiting now the authors to confirm that paper is, everything's okay, and we can be accepted. So it's one step before accepted. So this I could submit it. So we have the microorganism. You need to select appropriate microorganism. This could be yeast, this could be gram-positive, gram-negative. If they have their benefits, you need to culture them. This can be liquid culture, it could be solid-state fermentation, because you want a biomass, you want the cells. When it's question to culture them in the solid-state fermentation, you can use several waste products from the food industry or from the other processes. And you can actually use this solid-state fermentation applying from the very cheap substrates, and in this way you can have a cheaper way to production. When you go into a probiotic, you're aware a lot of possible contamination from the media, but in this case you can even combine all the media residue, all the cells, all the biomass, because if you are using the food residues for the growing, it's even, you can be much more easy for you. Then you need to find the way, you need to kill this microorganism. So remember the story, how to kill Listeria monocytogenes. Artificial intelligence say you're not supposed to kill. Well, we need to kill the microorganisms because we're going to apply them as a dead application with beneficial properties. So this process could be very different. Very often people immediately think about heat. Well, autoclaving, it's a kind of high temperature. We kill the microorganism. But then we're having a big problem because if you kill them by heat, it's what? Okay, if you kill them by heat, what's happening? Maybe you can destroy some vitamins, maybe you'll destroy some enzymes. So you're losing probiotic, you're losing beneficial properties. So very often, this way, this step here, how to kill the live microorganism and to prepare the postbiotic is a very crucial, very important step. Because you need to be sure that you're not destroying beneficial metabolites. So obviously, high temperature is the easiest way, but maybe it's not going to work very well. So we can use the high pressure, because high pressure will maybe destroy the cells, but at the same time, most probably will keep the metabolites in the good condition. You can use sonification, but well, sonification can actually destroy some of the proteins. And if you're having some enzymes, they're highly molecular weight proteins, maybe sonification will have some problems. Even in some situations, sonification can maybe even have negative effect on the DNA. And if you need a DNA or RNA to serve as an inductor for some beneficial process, maybe we'll have a problem. Mechanical destructions. We can always use the glass beads and destroy the cell by the highly speed vortexing. We can use some chemicals, so we can use even some enzymes. Idea very often to combine all these methods in the way, then we're going to preserve our objective, our metabolite that we want to have it in the postbiotic. So then additional biotechnological approaches for postbiotic preparation must be developed because obviously we're having the beneficial metabolites, but again, most probably we'll need to do some kind of modifications to remove some of the components and to focus on that kind of partial purification, but still not fully purified, because if we fully purify, we work with the metabolites. And then biological and chemical assays to need to be selected products with stable biological activity and chemical properties. So now, analytical chemistry needs to confirm what kind of metabolites are they and if they're having even quantification. So you make a postbiotic preparation, you say, okay, this postbiotic preparation has this level of the vitamins, this level of the enzymes, you need to do this part. But end of the day, you're taking advantage of the probiotic cells that but this is not obligatory. That could be not even probiotic. If they're producing metabolites, great. If you have the metabolites in the end, this is the point. And you can go far away of the problem that you need to keep the live microorganism. As well, the point, very important point, is the fact that for immunocompromised people, even the best probiotic could be dangerous. In this way, we're going away because we don't have nice cells. We are applying metabolites produced by these microorganisms. So, in fact, from the same paper, probiotics versus postbiotics, possible systematic infections. Even the best probiotic could be bad for some group of people. Colonizing, well, we want to put these cells in our host, but very often long-term colonization. What about later we discover that this colonization is not good? What we'll do with this strand is already there. In postbiotic, we don't have this problem. We have safe and more controlled application. So you're giving now, doing the job, you're giving again, doing the job. You don't need it anymore, you stop giving. Stability of these products is much more stable. They can be kept very easily and more. We know what exactly you have. In the probiotic, sometimes they can express something else. They can make attachment. When they're in the gastrointestinal part, maybe they will produce something different. Then we never think about this because specificity of the metabolic pathways can reflect and produce something different. Technologically advantages, formulations, delivery applications, all this is a very benefits. And this cartoon is like the probiotic, say to the probiotic DAY, but probiotic alpha. I have never been a probiotic, but even then I will be better than you. So they can work and they can perform better. By the way, don't say this to my chicken, because I hope she will not be watching this video, but this actually looking a little bit like him. He liked to wear this kind of spectacles, circle one. It looked like, but he didn't draw it. So maybe he was actually playing and putting me killing him as a probiotic killing postbiotic. Okay, then some of the metabolites produced by the lactic acid bacteria that can be actually considered as a way of the, now coming to this postbiotic story, GABA. Gamma aminobutyric acid, called as a GABA, can be produced by several lactic acid bacteria and in fact can have the beneficial properties. Well, some probiotics are already known that can express GABA even in the gut environment and they can be producing. But again, what about if we can have the control fermentation process? You have the strain, we're producing GABA in the biofermenter, and then kindly concentrate and slightly go rid of what we don't want, and we have kind of dead cells together with the GABA. So it's not a bad idea. GABA is an inhibitory neurotransmitter. It lessens the nerve cells' ability to receive, create, and send chemical messaging to other nerve cells. So now a transmission of the neural signal is very dependent on the presence of GABA. And many medical conditions are in fact associated with changing levels of GABA, and medications target the GABA receptor. So more evidence are needed to know how the GABA can actually modify. Well, in this moment, we believe if we add a little bit more GABA in our food products, and if you're having bacteria that can produce GABA, it's good for us. But again, with development of the research and small studies, we will know better about this. By the way, in Korea, they're selling chocolates with GABA, enriched with GABA. Well, I tried, I don't feel much more better, but they say it's a good for me. So, by the way, they're not more expensive, the same price without GABA, but okay, we can have it. What's wrong to have it? Okay, GABA is non-protein amino acids, so it's very important, this point. Go. Okay, normally it's associated as neurotransmitter, so the presence of GABA will facilitate transmission of the nerve signals, but at the same time for the bacteria, because GABA is important for us, but what's making good for the bacteria? Why bacteria producing GABA? It's actually helping them to resist to acidic conditions, because what's happened, come on, okay? When you have a high acidic level, bacteria need to deal with these ions. And what is happening? You're having glutamate in the presence of low pH, so presence of hydrogen ions. Glutamate is transformed to GABA. And in this way, in fact, you block the presence of H plus ions. What's the name?
Hydrogen ion.
Okay, because in Bulgarian it's different, in Portuguese it's different, in English it's different, so need to remember all these names. And normally this it's associated, production of GABA, in fact it's associated very much with one enzyme, GAT. So if you can detect this enzyme, it means the strain having chance to produce GABA. But now, we was doing this project as well to select some beneficial microorganisms that can produce GABA. So one of my students, he just defended his master and now starting doing his PhD. He was isolating strains that can maybe produce around GABA from Brazilian cheeses and from feta cheese from Bulgaria. So I brought this cheese in Brazil. It's allowed. It's not against the law. You can carry the cheese to Brazil. Dairy product is allowed. Meat product not allowed. And I was carrying for me to eat this cheese because I like it. But then when we start the project, okay, let's get the samples and try to isolate something. So first of all, when I was isolating the strain, we was checking for gram catalase because we wanted lactic acid bacteria. I think actually I can sit here. I think for the camera it's okay. You can see me. Okay, a little bit tired. I mean, 5 days, it's a marathon. Okay, so we was doing gram and catalase because we wanted to be sure that we're selecting lactic acid bacteria. It's easy. I mean, catalase gram, okay, gram take a time, but take to observe the strains, but I mean, students need to know how to do the gram. So we selected always lactic acid bacteria based, okay, presumably lactic acid bacteria. And then we wanted to know if this bacteria can maybe produce GABA. The method, someone already worked with the GABA production here in the room? I'm suggesting you to do this work. It's not difficult in generating quickly results. So in a very lazy way, in not too much work, you can have a lot of results and impress your supervisor with going on the meeting every week and showing a lot of results to easy to generate. So you have the samples. Half of the samples we centrifuge because we need to obtain the cells and supernate them to separate. And then we use the pellet. On the other side, we take the samples, we sonificate them because we want to destroy the cells, to liberate if something is inside the cells. So two options. Then two samples, sonificated, sonificated sample and the normal pellet. And we use the spectrophotometer method to do this test. We use an ELISA plate, 96 post plates. And then a very simple method when you add buffer, bronchresol, pyridoxal phosphate, and then you add glutamate. Because with this way, you're going to have glutamate as a substrate for production of GABA. So if you have presence of the enzyme, GAP, it's called GAP, you're going to transform glutamate, and you have the indicators, they're going to have a change in the color. So you put in the microplate reader, and you start reading every minute, every 2 minutes, because if you have this kind of line that's going up, it means you have reduction of GABA. It means the enzyme is there, is transforming glutamatic acid to GABA. If the line is like flat, no. Well, in this project, we had to acquire a big number of strains, and we was trying to pre-select some of them. Again, more or less about 400 isolates from cheeses, but on one plate you can add 96, okay, you have controls, let's say 90. So make 4 plates only, and you have the results in, let's say, in less than 2 hours. So, okay, he never done in one day all the work, but I mean in less than 1 week, he was able to see which of his isolates, in fact, have the potential for GABA production. But since we're always looking for bacteriocin production, he was as well checking what of these strains are bacteriocin producers. So after that, he crossed the data of bacteriocin and GABA production, and he selected 8 strains for the future study. Well, he selected a little bit more, but then do the DNA extraction, do the red PCR, do the identification, and realize that some of them are replica stem strain isolated in multiple occasions, but in the end, he has 8 strains that go further. Then question is how the GABA production is dependent on the condition. So we tried one of the time methods. So you can play with different cellular density, but you keep all the other 4 parameters blocked. So we have it as a standard 10 to 6 cellular density, pH 6, incubation time 24 hours, monosodium glutamate at 60 millimolar, and temperature 27. So if you change the cell density, all the other you keep in the same. Then you keep just play with the pH, but you keep cell density in the other constant. Incubation time, you do it 24, 48, 72, 96, 120 hours. But the rest of the parameters, you keep stable. You don't correct. Then monosodium glutamate is mean the precursor for production of GABA, and temperature. So what we observed, if you do this variation, you can see that the GABA production is very dependent on several conditions. So let's say you play with the sodium. Okay, this is the different strain when we selected, and this is the different conditions. So you see, in some conditions, sodium glutamate is very important. In some conditions, the time of incubation for other strains is as well one of the important moments, or temperature could be playing as well reaction. For some of the parameters, you can see it's a different condition. So obviously you don't have the same conditions for all the strains. Again, it's a bad news, you need to do it. And the problem of this process is associated with a little bit more work because all this process needs to be done on the mass spectrometry and partial purification of and quantification by the MSC mass spectrometry method. So in fact, if you do kind of generalization for this 8 strain, you can see that more or less 10 to 6, it's more important cell concentration that's important for GABA. That actually is good news because it's a little bit below requirement for the probiotics. So if you apply these strains as a probiotic, they have a good chance to produce GABA in our gut. As well, when you look for the pH 6 neutral, it's a good news because imagine if it was 4, it would be a problem because I mean for our gut system, it's lower gut, it's not 4, there is like neutral, so it's a good news. And as well, glutamate concentration needs to be quite high for this optimal production. But well, glutamate could be delivered by several food products because it's a part of the food, so no problem to deliver this. We don't need to give glutamate, pure glutamate. We're going to give food products rich of glutamate, but this way we can actually have this production. And then time of the incubation could be for 24 or very long. Well, most of the time food stay in our lower gut about 24 hours, so time enough for this mechanism to produce GABA in silico. Well, temperature, 50 degrees Celsius is the best one. So it means if you're going to biotechnological production, it will be possible to do a little bit higher temperature because you put in bacteria in stress, very high, not going to kill the bacteria, but it's a stress, so obviously pushing them to produce more GABA because most probably the pH is trying to affect the strength. But again, the 37 was not bad at all. We have in the second peak on 37. So all this showing that the GABA production could be done in silico. And then with GABA, now we wanted to know in the next project what going to Kaiki or Donio, she just started his PhD like a few weeks ago. He was actually wanted to see how the GABA could be applied, and he wanted to use the mouse model to see because the GABA can actually have an anti-cancer effect as a component. And since some of the bacteria that he work is showing a very good anti-cancer effect, bactericin himself. So now he wanted to use GABA and the bactericin in combination and use it on the cell line and after that on the animal model to see if he can manage to control some of the gastrointestinal cancer that could be effective, could actually be controlled by this combination, bactericin and GABA. And he's going to explore this on the molecular and physiological and animal model because idea is to having this probiotic later for the prevention because these days we can detect possibility of gastrointestinal cancer by some markers. You do the blood test and you say, okay, maybe you're going to develop this kind of cancer because marker is supposed to move. And for these people, they still don't have cancer. They're clean, but they're at the risk factor. So what's the best option if you can apply the probiotic that can have reduction of this cancer and you can actually prevent them on developing of the disease, or if at least you can push it like, I mean, in this case, even if you push 10 years later, it's even better. Because if you can prevent, it's excellent. If you can postpone the disease for later, it's another option. But again, it's prevention will be a good option. Another project that is run, we are testing different fermented food products that are normally consumed in Brazil. You see the variety of them. Some of the names may be familiar, some was completely strange names. But well, there's a variety of the fermented food products, and we were screening all of them for the potential bactericin producer and as well potential producer of bioactive metabolites with the idea to apply them as a probiotics. Interestingly, we have several very strange, lot of bacillus beside this isolate with a good antimicrobial production. Some of them were already tested and we showed that they have probiotic potential. Even on the cell line, there was showing increase of the anti-inflammatory cytokines. So it's showing a good future and potential. Well, this work is going by Emilia. She is a postdoc in the laboratory. But the problem in Brazil, the postdoc, they hunting the job all the time. Because the postdoc is for 1 or 2 years and you want a permanent job. So she hunting the jobs and doing the work in the lab. So in the moment when she finds a contract and moves to a permanent job, she will leave the lab. But again, there will be another person, maybe some of the PhD will continue with this work, and we're going to complete and see which one of these fermented food products, in fact, could be more promising later to be developed as a functional food product, because we're going to enrich them with their own bacteria. And since we know what is the beneficial properties of this bacteria, we're going to know how we can apply them and what we can use them for. Well, it's everything just the angels. Now it's coming the problems with the probiotics. Well, according to the probiotic, first about the probiotic and fermented food products. Very often people say fermented food products are probiotic, fermented food products are probiotic. No, they are not. They're beneficial, functional, fermented food products. They can carry several microorganisms with beneficial properties, but they cannot be considered as a probiotic because, first of all, we don't know what kind of bacteria we have there. We know the product is good, you're feeling better, you're having stomach problem, you take these fermented food products, you feel better, but why? No one's knows. The problem is probiotic needs to be fully identified in medical clinical study proving these beneficial properties. In fermented food products, in Daki, you have nice bacteria, but you know what they do exactly? I do identify them? No, they're there, they're good. We know in general they're good, but they cannot be called probiotics. Like kombucha. Who like kombucha to drink? Well, it's strange fermented tea. Chinese like it. They're advertising a lot, but it's not bad, but still. There's several beneficial microorganisms. Some of them are lactic acid bacteria, some of them are acetic acid bacteria. They have several E's. But again, we don't know the identity. And we know their number, exact number. To be a probiotic needs to be reaching us with specific number. So taxonomy not identified, not scientifically proven specific beneficial properties, so neither confirmed by animal or human trial studies. And the presence of appropriate levels viable cell, all these things actually clearly stating that fermented food products cannot be called probiotic. This is why several dairy industry, they stop actually advertising their fermented food product, yogurts, etc. as a probiotic, because this is a specificity of the point. Even if you look here, if you make this kind of study and you put the kombucha and see what people associate with the kombucha, they say it's a probiotic very often, but we see functional food, probiotics, microbiota, health, bioactive compounds, antioxidant activity, green tea, organic acid, polyphenols, phenolic compounds, all these things. But this word, probiotic, cannot be linked with the kombucha because kombucha is not a probiotic. You can make same kind of analysis online, and you can generate the figures for any functional food products, any fermented food products. You will be surprised that several papers still associated fermented food products and claiming that they are probiotic. But it's not true, it's wrong, because they don't fit the definitions. What about the other side of the story? Several cases, probiotic products on the market. You see, till now was like a wrong perception. The industry even sometimes playing and they climbing the probiotic, something that is not probiotic. But now you have the pharmaceutical products that you buy even in the pharmacy, and they say it's a probiotic. Okay, thank you very much. They have the clinical studies that prove this strain, specific strain. In fact, it's a good. The problem very often, these pharmaceutical products, they contain a wrong strain. Someone was working with commercial products and mentioned that very often they isolated different strains, different species, not strain, even species. Sometime on the label they say it's a single, well, most of the time it's single culture. But you start isolating, you realize there's 3 or 4 or 5. You don't not even to do the sequencing because when you observe the colony, when you do isolation, you realize they have 5 different type of colonies. Obviously, it's representing 5 different strains. This is one of the biggest problems. Very often, even the good quality company sometimes can happen to have the wrong, uh, not more than one or even the wrong strain. There was a case, I'm not supposed to say the name of the company, but neither will say the country where this was happened, but probiotic on the market, in fact, was contaminated with salmonella. You want to have it salmonella? Yeah. So you think that you taking something that is good for your health and then you're having medical more serious problems. Containing low number of the probiotic lactic acid bacteria, not only lactic acid bacteria, it can be yeast, can be new generation. Very often because of the wrong packaging, very often about the wrong storage, very often because of the wrong production, commercial probiotics can have the low number. And again, it's not fair. I'm paying a quite expensive price for these probiotics, and I will take it, and then I'm having nothing. One of the studies that was performing, every probiotic was cost about $40 every package. Imagine you buying this and you take it, you realize it's not appropriate number. Containing dangerous bacteria, including potential human pathogens. This story with the salmonella inside the capsules. Well, the company actually acknowledged they had a problem in their production line, and they asked us to make this check what they have it. Well, in a couple of days, you say you have salmonella. So there was stopping the entire production, cleaning the line, doing all this. But I don't know if they remove everything from the pharmacies. I don't know. They're supposed to be, but who knows? Sometimes probiotics containing nothing. It's happened. We was checking some capsules, nothing. Like a sterility test, nothing available inside. One probiotic that is good for me maybe is dangerous for you, or one good for you is dangerous for me. Sometimes missing this information. They don't say who can take, who cannot take. Sometimes it's even not testing these things. And commercializing something like a probiotic that is not at all probiotic. There was even some opportunistic companies, they putting on the market, even nothing. They just put empty capsules and say it's a probiotic because they can sell it. Well, regulation in several countries is getting stronger and stronger. Let's say in Brazil, in order to commercialize a probiotic, you need authorization from state agency. You need to provide all the portfolio. They're asking to have the full genome sequence. They want to ask all the clinical studies, animal studies, everything in order to allow you to commercialize this microbial culture as a probiotic. They're not going to allow you. But again, remember I was telling you this story about the salmon and contaminated with salmonella. Some countries, they cannot sell their probiotic products in their country. They just export in the country that is no regulations. And this is not fair because, I mean, you're playing with the health of the people. So be honest and produce the right product and deliver this product to the people in the right way. Again, checking for some probiotics for same species, we realized that even on the label was written that it's the same strain in the 3 different products. We obtained a species that it's okay, but obviously the strains are not the same because red PCR was showing they're different and they cannot be the same species. So it's not the same strain. There's some classical papers on this showing analysis, and this is like 20 years old paper, but clearly stating that several probiotic products don't match what is on the label, what is inside. It's getting even continued. 20 years later, we have several examples that the products on the market was not really corresponding what do you have inside. Then you're having this dairy product. It's okay. The company is already not on the market, so we can talk about them. They changed their name. They was providing this kind of dairy. It's like a yogurt with fruit juice. It's a very nice and tasty. It's refreshing. They was having with different options of fruit juices. But again, if you look, I hope I can manage to do this. They have the simulating functions of the immune system. They're climbing this on the label. Uh, they playing well with the lactose digestion. For the people that with lactose intolerance, and well, the product you can easily see from the website is from South Africa. Africa, they are quite problem lactose intolerance. Inhibit the growth of harmful bacteria. So what do you understand by harmful bacteria? It's not really clear. And then clearly stated contain probiotic culture. Well, everything sounds very good to be true. So I was enjoying this drink, and I decided let's check what kind of probiotic they have inside, because my idea was to use this culture as a control culture. Well, bad news. No lactic acid bacteria presence in this product. But we have Staphylococcus aureus growing from this product. So you see, they claim that they have the probiotic. They claim several good things that can happen with this. This is taken from their website. But again, I think it's not anymore existing, this product on the market. Neither the company is existing anymore. So there was claiming several beneficial properties, but at the same time, there was not having nothing. It's like playing with the sense, because the people see this probiotic culture and they will buy it. And very often it's more expensive than normal other products. So it's again something that I'm criticizing the system that, okay, now let's keep it this quick. Sorry, it was Indian company. They get the problem with their probiotic properties. They were selling this in South Africa, but after that they discovered that there was some problems. They were taking really good action to remove the products from the market. So, but this is like 20 years ago. These days, the company needs to play more powerful, because they actually need to be sure, because the customer is much more aware of their right. They know much more better what is product. They know much more better how we need to control, how we need to play with it. Because if not, you can having a really problem. Customers calling and asking, you provide me something that in fact is not working very well. Many commercially available products are incorrectly labels. We're supposed to label the probiotics in the right way. Regulation says not only to put the name of the bacteria that is inside, but you need to put even the strain. Because again, sometimes the regulation agency, they authorize a specific strain. They don't authorize the species, they authorize specific strain. Frequently, bacterial count is not corresponding to what is on the package. Some products contain no live bacteria, they cannot be considered as a product. Well, if they have the dead bacteria, maybe you can claim them as a postbiotic, but not a probiotic. And then some other strains from the same species is not a question. If you say you have this strain, needs to be this one. Then coming to even worse, sometimes they claim something as a probiotic that cannot be probiotic. Please try to convince me that you can keep the live bacteria inside the soap. If you convince me, okay, I agree. But I don't believe you can keep it live bacteria inside the soap for years. Neither in the lipstick, neither in the shampoo. Well, very often this could be dead bacteria. They even deodorant with probiotics. I mean, how you can keep this? Imagine this bacteria in this under the pressure inside the bottle with all these frills and all these things. How we can manage to do this? I don't think will work. Then another problem, uncontrolled application. Now it's making a parallel with the antibiotics. Antibiotics was a good intention, but they generated antibiotic resistance because of the uncontrolled application. Maybe uncontrolled application of probiotic will generate some problems in the future. Well, we already removed this from the dairy products and from the several fermented food products. We already not allowed to commercialize them as a probiotic because they are fermented products. They are beneficial, they are traditional. They could be functional, but they are not probiotic. Already it's kind of moving, but we need to be very careful as well when we suggest new probiotic to be really sure that we not creating new Frankenstein, because putting the bacteria that can be colonizing very well after that, if we need to remove, how are we going to remove it? So all these things is very important. And then remember that the industry like to play with the sense of the customers, because you're having this kind of spatial, buy 4 million day, you have one for free. Well, it's nothing, you have nothing for free. But again, very often the word probiotic or friendly bacteria will catch your eyes in the supermarkets, and you can go for something to have it even pay more expensive, and because just of the word, playing on the sense of the customers, and in this way you can actually improve yourself. But again, everything is need to be controlled and to be know what we're giving. As a responsibility of the science, we need to know what you're suggesting to the industry to be applied in order to don't have problems later. So, okay, you cannot see the text, but I can tell you, don't be afraid to be innovative and creative. Never. Just remember that Art of Noise was built by only one person that never been to the university, never have the engineering studies, and the Titanic and Costa Concordia been by the professionals, but this one survived, these two go down to the bottom of the ocean. And this is actually the acknowledgement for all my collaborations that I had in the past 30 years. So even Wade the Santos in this list here. It's quite a good collaboration with him in the last more than 10 years. I had my study in the Europe, in Sofia University. Then I was working in Inicia and Indra in France during my PhD, collaborating with the colleagues from Lille, Germany, Portugal, Slovenia, Italy. That's from Africa, Oran University in Algeria. Then I was actually in South Africa for like almost 6 years, working as a visiting professor. In Korea, in Kandung Global University, collaborators from this is Hong Kong, Malaysia, Philippines, New Zealand, US. We have Rutgers, Mike Chiquindo there, Harvard, UC Davis. In Latin America, is several: Venezuela, Colombia, Argentina, different Brazilians. And in fact, what I actually wanted now, because this is my personal collaborators. I want this one to become a university collaboration, and I hope you work. And this is actually the students that work with me. It's a present time we have this team. These students been already in the lab, they left, they go to the different places. This one is coming after taking this picture. And it's a fact all this work is because of the collaboration that I had around the world and the team that is working with me. Do you want to get? Okay. Do you want to have a break, or you want to go for what's the rules? Can we have a break and then we go for the discussion? Okay, break.
We continue interaction. So पानी पीना तो पी सकता है. Keep on interacting and it will take half an hour to finish.
So we're going to continue, or we're going for the tea, coffee, water, walk? You're the local boss, so tell us what we're going to do. Continue. Okay. So what's your opinion? what. Tell me what you want to hear more, because sometimes I don't have the feedback, and in the future when I do the course in another place, based on your feedback, actually I can move the slides and move the topics and use it in the other situation. If you ask me what will be the question of the test, I'm not going to answer, because I still don't know. I need to write these questions tonight. Or you're so tired of the bacterial scene, lactic acid bacteria, probiotics, postbiotics, metabiotics, mortobiotics. There was actually, it was kind of joke. They wanted even to call in one moment mortobiotics. Because, I mean, they're dead, but not sound very good. I mean, imagine you put on the label of the product, this product contains mortobiotics. I mean, it sounds dead biotics, it not sounds very good commercially. The problem with the story of postbiotic, we don't really have a good definition in the moment. There are several definitions. Different organizations propose it. When it's questioning about probiotics, World Health Organization, they're having like consensus and have kind of generally accepted definition. When it's coming to postbiotics, there was some kind of organization they suggested, there's some paper they suggesting, but still no governmental or international body that is actually having consensus what could be. So it's a very speculative area in the moment. Some people even consider postbiotics some metabolites, but in the general perspective, not supposed to, because it's a metabolite. If you isolated an enzyme out of the culture and you use it, it's an enzyme. Why should call it postbiotic? So, in the moment as a postbiotic, it's considered something that is non-purified and entirely dead cells containing metabolites. But if you purify, you have bacteria, you have enzymes, you have polysaccharides. In case you're applying everything, it's considered as a postbiotic. So there is again some adjustment in these days. Really, it's if I give a definition for the postbiotic today, maybe tomorrow will be changed. Because again, no, if I think World Health Organization or FAO, it's Food and Agriculture Organization, they need to come with some kind of decision what if in fact postbiotic could be. There was some even kind of fight between the groups. One group proposing something in the paper, another group answer and tell them, no, you're wrong. Then the first group again gives the answer, another one attacking again. So it's a kind of different research group have different opinion. But again, very often the truth will be born after the long discussion. Questions, opinions, critics?
इस सेशन का क्वेश्चन डिस्कस कर लो उसके बाद we are inviting director NTSC local coordinator of this program who will join us at 3:30 and we will have formal feedback and concluding session. Meanwhile, we can discuss any issue related with your personal question, discussion, question related to the whole country, like whatever the lecture you had. And not only to the question, but you can ask which encouraged you from UG, PG. All UG students, how you find it? Just sleepy? Initially they were interested to join, but I think after the course they thought that it is useless. Because I paid 800. Come on. You learn something or not?
In our conversation in the last couple of days with the authority of the university, the vice chancellor was giving very good signals that he believed that deeper collaboration between University of Sao Paulo a new institution is more than welcome. He was clearly showing that this is a good idea and needs to be explored. So well, now it's coming the question. We need to do the work. We need to establish this formal part of the collaborations, sign the appropriate documents, because all these documents in collaboration actually protecting you. When you go to another institution, if you have the agreement for collaboration, you're better protected. It is important. And then you can do your research as a sandwich method. You can do a short visit to that institution because it's important to have the touching the different institutions, different cultures, because this will enrich your expertise. And the idea is maybe we can start together with Professor Tivari research group in the beginning, then extend to the other research groups. Because I mean, what's better of this to go and to spend some time in another laboratory, having some complementary expertise on your project from another labs, and maybe later some postdocs in another lab. Even maybe we're going to think about the option to have this double degree. It's another possibility that exists in these international relations. So you make a part of your PhD in here, another part in another institution, then you have two documents for the PhD from the two institutions. This kind of agreement is completely working in the last few years. It's getting more and more popular, giving the better options for the market on the when you look for the job and positions of the universities. Well, it's a lot of work to do this. Do you don't think? You think we send these documents in 5 minutes? Lawyers need to check all these documents. Lawyers here, lawyers there. They need to be sure that nothing is against no one. So everything to be mutual agreement and mutual interest for both sides. No one is more than the other. In this kind of agreements, we need to be equal, both sides. No one can claim that is better than other.
So what he's suggesting that we can do at both levels, and the PhD students, the benefit for the PhD students that they can have a student exchange program under the MOU. The university from both the sides are agreeing to sign the memorandum of agreement for the student exchange, for the faculty exchange, for technology sharing, so that it can be benefited. Till that time, we were only connected to understand what are the role he is doing, what I can do in my lab. But other students from any field related with him, if he is working, or any neighboring department, if it is working in his university, that can be mutually shared and understood with the concept. So, like a UG student, if you have completed your UG, you can think of doing master not only in the Sao Paulo, if it's possible, or if you go for anywhere in the master degree, you need a recommendation letter. That Professor Rohtak will be getting the recommendation letter in other universities as well. If you are interested to join that university, our university can have a MOU and do that sharing program. And master level student, if you complete, if you want to join PhD program in the Sao Paulo or related university, that can be. PhD students always lack the techniques and the high-end resources. It is not available throughout the world. Some lab like this technique, some lab like other technique. So there is no lab which is full of technology or anything. Everything is not fine for all the lab. So we need a kind of complementation, that complementing the kind of expertise what this lab has, and the kind of work which cannot be done in this lab may be done in other lab. So you need that kind of one or two or three network which can at least complete the total work. In the research, it is not possible to maintain the laboratory in any one of the country, not in only here or Brazil. So this is the reason the platform for networking is important. What is the course we have this global initiative for academic network. Why this government initiated this program to have a network of academic level or research level. Only they realize on the theme that we cannot do in isolation. Any country or any researcher or academician, if they are doing alone, it is not possible for them to do everything. So you need a collaboration with the hospital, you need collaboration with the industry, you need a collaboration with the one university with other university. Some universities are performing better in the STEM area, some university performing in the humanity area and other traditional knowledge. Then that can be possible only through the collaboration and understanding what you can do, what others can do. So this is what our emphasis was, that how we can apply for this proposal through the Gian, through the ministry. And if someone is coming in this course, he or she may give you the guidance which we are not able to give. Probably at that university there is a limitation in terms of shortage of faculty or their experience in the faculty, what they have, the kind of exposure of the faculty. That is, again, uh, we have this technical limitation everywhere. So the government thought of this. And this is a kind of opportunity for you. You realize that thing because you have preferred the course uh, before holiday and the vacation you have. I think it is a great appreciation for the participant that they have choice for the course rather than vacation. On 20, after 14th, they had a vacation. And I think you can enjoy your vacation after. Tomorrow is not a problem. Tomorrow we'll have only 1 hour from you 10 to 11. After that, it is yours. 3, 4 days you enjoy. You did something which was academically correct, and that way you can do much better. What you learn that can be helpful for your future discussion. And yes. So out of this coverage, why I'm telling this story, that if you hear all these things, understand what is your opinion, if you want to say anything, if you want to interact with the foreign experts.
Even if you look at what's the name of the program, it say International Academic Network. So my job was not just to talk to you here, but as well to try to establish more deeper academic and scientific collaborations. Idea is to make a huge network with everything possible because, well, I'm presenting Sao Paulo University, I work there, but as well I have my contacts with other countries. And, well, we were discussing some projects with Santos Tivari. Well, okay, I cannot do this in my lab, but I know someone in South Africa that can do it. So we can maybe send a student there. Okay, but now carotenoids, there are people in Russia doing with this. Okay, let's send a student there. Okay, but another one work very good on this area in France, in INRA. Okay, could be there. So actually generating this network with the options and see expert, what we can actually profit of expert of specific area in the specific country. Why are you so shy? No one wants to ask nothing. Okay, good. Give it the mic.
So, can we genetically engineer probiotic strains to broaden their spectrum of bacteriocid activity or improve food safety?
Again, we can do the genetic modification. This is well-known already technique. You can sit and listen. The problem is we can do the modification and use the strain in the biotechnological production of the bacteria. But if we wanted to apply this strain as a starter culture or in adjunct culture, we're having a problem. We cannot even apply it as a probiotic because it's GMO, genetically modified. And according to regulation, we're not supposed to apply genetically modified. But again, for the biotechnological production of the bacteria, it's no problem, they do this. The problem is when we do the genetically modified, we need to as well to treat in the specific way the strain to keep the genes to express or express, and then these strains could be a hazard. We're not, we, at this moment it's not allowed. Something that we can do, it's a kind of tricky way to go. We can do the natural generated mutants. You can expose the strain in some conditions, so you can do the modification, natural mutants. You can generate natural mutants, and then you can select these natural mutants and you can use them. But if you do the genetic modification, like cloning, adding, modifying the genes, unfortunately, this is a good academical exercise. It's a good thing for the industry to produce it as a biotechnological metabolites, but not allowed to keep them in the nature, giving them to the people, putting it as a starter culture. It's just the safety issues. I think we're still not as our knowledge and our society is not yet ready to explore this, and rather have this precaution because we don't want later to regret liberating something that could be some negative consequences. You see, I'm a little bit skeptic about this. Even genetically modified plants in the nature, I mean, could be some negative consequence. There are already some negative consequence. I don't know, in India, if you use the genetically modified plants, are you need to mark this on the package or not? Because in Brazil it's compulsory. If you have this letter T on the package, it's meaning some of the components in these food products are results of the genetically modified plants. We need to annul this. And in some countries, they say you cannot give this kind of products to the children or to the babies because, again, for the safety precautions. But some countries, they don't have regulations. You can, I mean, I'm afraid to say this, but maybe most of the corn produced in the world is genetically modified these days. So if you need to cut genetically modified corn out of the market, will be like, what kind of corn we can have it? The problem, you give this to the animals to eat, then you eat this meat, and could be some, well, they made it study, they was climbing, that is safe. But again, it's, at least with the microorganisms, we are still keeping this regulation. We're not allowed to liberate them in the, as a probiotic or as a starter culture. Okay, in the production of the bioreactor in the industry, yes, but they're controlling to don't release them in the nature after that.
So it's Indian regulations are very tough. They are not given the safety clearance for the release of these varieties. Even in the plants, people have done that experiment, but they are not getting considered environmentalists from the clean.
But the big companies, they actually providing seed material that's already genetically modified. And it's, I mean, they claim that they made it all the studies, but again, I actually am very in favor to governmental decision to clearly control this because it needs to be controlled. If not, will be house. If it's not controlled, will be really end of the world. It's much more philosophical discussion in this point, but again, yes.
Sir, how are bacteriocins are useful in cancer treatment?
Well, as I showed you some results, there were several examples that can be inhibiting only the cancer cells, but not healthy cells. And this, you can now correct me, Cower? No, it's not Cower and Cower. How you pronounce this too? Core and Core. Okay. There was really one of the pioneers showing this in systematic way. They made this review paper. There was putting attention in this topic because several papers was climbing this activity, but no one was putting together. So it's actually a very good point because after they started more and more people working in this area. So we now know that the bacteria can recognize because of the specificity of the surface of the cancer cells, the fact they have much more negative charge, they have some demodification, and the bacteria can recognize them better and they can attack them. Uh, Several works now showing, but the question still nothing on the market as a product. The academic research showing that bacteria can attack cancer cells and can destroy them. Again, there was a kind of worry, what about if the cancer cells are inside in the brain or another place? But again, pharmaceutical industry already have the methods to deliver some metabolites to the specific place. This is not a problem. If we're talking about the skin cancer, we can even make a topic application. Well, if this is our body need to have this specific target polymer that will help to deliver to specific place, and in this way we can attack them, because the good point, they don't attacking the healthy cells. This is more important. Chemotherapy, they are attacking the cancer cell, but they're attacking the rest of the cells as well. As I mentioned, they destroy your gut microbiota. It's so aggressive. In this case, you apply the probiotics that can be restoring your gut microbiota to help you to fight with the cancer. Probiotics, they are not going against the cancer. They're helping you general health status to improve, and in this way, you're better in this fight against the cancer. But with the bacteria, since this is the idea, they're recognizing, they can interact, they can destroy cancer cells, and in this way, they will improve. Most probably not going to be only single treatment. Most probably will be combined with some chemotherapy, most probably with some other methods. But again, in such a complex disease like a cancer, we need to think about the complex approach. Any option that will improve the status of the patient is more than welcome. You can do some project on this if you want in your next study, because it is what can be done. Yes?
So we know that probiotic bacteria can have some limitations or some problem, as you mentioned. So can we modify the probiotic bacteria, not genetically, but with any other technique or any other method? We can modify it to have the same advantages, or just to overcome its limitation as we have in postbiotic bacteria.
Yeah. Well, easy way, you can do the selection of clones. You grow your strains and you're looking for some specific benefits. And when you have your strains, natural mutation is something that can appear. You can actually induce some mutations. You can use some simple methods to generate mutants. And you do the selection between your clones, the more important, more stronger, most one that is work better. Even some people say you can learn your strain to live in the higher temperature. You can learn your strain to live in the lower pH. I don't like to use the word learn, but again, if you constantly expose your strain to the lower pH in the conditions that not going to kill it, but will most probably the strain will try to find a way to adapt to this lower pH. You see the GABA production, it's for us, it's neurotransmitter, but for the bacteria, it's a way to escape of the low pH. So if your strain have the gene for the GABA production, and you expose it to the low pH, you will start expressing this gene. Maybe even overexpress this gene because it needs to protect for the low pH. And you, in fact, selecting the strain that will be more resistant to the low pH. So this strain, in one moment, will be more effective when go via your gastrointestinal system, because you have better chance to survive the low pH in the stomach. So if the strain having opportunity to express some metabolites, in this natural selection process, you will pre-select the clones, mutants, natural mutants. There will be better survival conditions, and there will be better way to copy with this hard condition that he needs to survive. On the other point, when you work with the probiotics, it's always good, actually not always, it's compulsory these days to have the full genome sequence and appropriate bioinformatic analysis, because you need to be sure what you're putting on the market. When you have the full genome sequence, actually, you can detect if there is any genes that will give the better survival in the low pH, in the presence of the ox bile. If he can produce some enzyme that can destroy bile activity against bile acid, this strain you can actually go and try with these conditions to put it in to generate natural mutants with a better expression of these genes. Well, if the strain don't have nothing related to GABA production, obviously you cannot generate this process and to improve the stability to a low pH. But if you detect in this bioinformatic method, you detect in the full genome sequence specific genes, okay, I will try to generate the mutants with natural mutants, of course, natural, not cloning. And you can try to push the bacteria to express better this condition, then you give it better possibility to survive the hard condition. Or better to express some metabolites, because you wanted some production of some metabolites that associated with antimicrobial activity, associated with production of short-chain fatty acids or other. But if they don't have genetic material, obviously you're not a magician to do it. But if the gene is there, you can put it in some pressure, environmental pressure, stress pressure, so you can actually push the strain for the better expression of this condition of your particular interest in your work. But again, please don't clone something and put it in the nature. Respect the rules and respect the nature. You can clone, you can do it this work. It's nice academic exercise, but this is for the laboratory, not out. Yes.
Sir,
Can you please explain the ability of probiotics to produce biofilm?
Well, probiotics, like other microorganisms, they can produce biofilms when they make this agglomeration and biofilm formation, in fact, could be a very good option for the probiotics that needs to colonize our gastrointestinal tract. Biofilm could be very bad things, could be very good, depends on what the biofilm is made. If you have listeria, obviously in the food industry, biofilm of listeria, you don't want it. But again, microorganisms go in our gut. They need to perform their job as a probiotic. They need to stimulate our immune system. If they're just a single cells all around our gut, they're not going to work very well. So if you can having this extra polysaccharides with these strains, if you have this addition properties, addition proteins on that surface, there will be better chance to make a clumps and form biofilms. From one side, you need addition proteins in order these strains to attach to the gastrointestinal cells. And if they can do the production of this interaction surface proteins, they will help them to make complexes. And once they generating this biofilm in our gut, there will be stronger adherence and stronger presence there. In this way, they will be much more better in the formation and consistency in the gastrointestinal part, and of course performing their properties. The problem with the biofilm could be actually bad news of the forming biofilm. Very often the cells that in the bottom of the biofilm, they can die because they have limited access to the food ingredients, of course. And again, if you make a biofilm very complex, maybe this can have the negative consequence of presence of the dead cells. So biofilm formation could be very carefully considered if they have biofilm formation. But again, in our lumen, in our gastrointestinal tract, they cannot be able to much to, because in the natural process, they will try to wash the dead cells there. So in fact, good surface proteins, good interaction, and some exopolysaccharides always a good idea, because they will help them to form the biofilm and to have the better persistence in the gastrointestinal part, if you want them to be there. Because if you remember the example with the heavy metal mycotoxins, you want them to come, catch the toxin, and go away. Actually, there's some idea to doing some kind of probiotics that can help in the reduction of the toxins from the food poisoning toxins. But again, it's a little bit, in this moment, classical method for removing. It's much more well applied, because if you start to giving a probiotic with a person with staphylococcal toxin, I mean, already process is like 1 or 2 days. And to have the effect of the probiotic, you need 1 week. So it's not a really kind of logic way. But for some other toxins, when it has a risk to be exposed to some food poisoning toxins, maybe it's not a bad idea to have it as a prevention for people that maybe will be exposed by chance to higher risk of these intoxications. I saw some microphone was going to another place. Okay.
Fish flow applicable to all the microorganisms, or there are some limitations?
Can you say again?
There is a concept you told about me that fish flow applicable. This is applicable to all the microorganisms, or there are some limitations?
Well, microorganisms, we're having our gut microbiota. We're having something that is normal to our gut. Our gut microbiota will change with the time, naturally. Babies, one microbes, young people is another. With the time, microbe is changing. But again, probiotics, we're putting something extra. So we need to be very careful what we put, and obviously a microorganism that is normal for the babies, maybe it's not a very good idea to have it in the adult, and opposite, because all these microorganisms, they have their roles of the normal functionality of the organism. And if you mix too much, maybe it will be something strange. Remember, I was mentioning for the bifidobacterium, it's excellent for the babies because of the specificity. For the adults, we need to be sure that it's not in the form that can be developed and then can work in our mouth. It needs to go in the gastrointestinal part. So again, it's a challenge from the industry, production. We need to know what we want, where we want, and how much. Because all of this is a complex. It's not just something that works in me, cannot work in your baby, cannot work in your grandparents. It needs to be appropriately designed for the specific groups, for the age, for the purpose, diseases to be taken care of, what we want to target, how we can improve this. Because again, remember I was giving these examples. Probiotic that producing hexapolysaccharides is not good for people with diabetes. It needs to be not given to them. So everything is much more complex and needs to be carefully designed and look from the multi perspective in order to be performing in the right way. Yes.
What are the key genes which are involved in synthesis?
Can you take a microphone, please? Because it's difficult to hear. I'm getting cold and not think very well.
Sir, what are the key? Sir, what are the key genes involved in synthesis and regulation of bacteriocins in probiotic bacteria? So, what are the key genes involved in synthesis and regulation of bacteriocins in probiotic bacteria?
What are the factors which regulate the production?
Okay. As mentioned, bacteriocins, you can see, bacteriocins are ribosomal produce. Very often, operon for the bacteriocin production is very closely related, in very often the same reading frame with the carbohydrate metabolism. So normally they're constantly primary metabolite. When the bacteria start growing, it needs to metabolize sugar. Well, needs the energy, no choice. And very often you read this bacteriocin operon, you will express the bacteriocin, normally the reasonably good quality. Because the bacteriocin is not just a simple killing factors, they involved in the conversation with neuron sensing and neuron catching with other microorganisms. So the constant production is something normal. When we are looking for the industrial application, we want them to produce more and more. So normally we can stimulate by some factors. Very often this can be different proteins. They will attach to these kinase enzymes. They will stimulate reading of these genes and the expression of the genes. So when bacteria realize it's some enemy around, this means these proteins from Listeria, they can serve as a factor, they can open the expression of the genes. Well, in some industrial applications, now it's coming to the kind of cloning. You can actually multiply the operon of the bacteriocin. So instead, when you start reading the glucose metabolism, expression of the enzymes related to the glucose metabolism, they will start actually, you multiply the same gene several times. You do overexpression. But again, this is genetically modified. You can apply it in the biotechnology for the overexpression, but not in the probiotic. When you have the probiotic, you need to do natural selection of the strain with a higher expression, with a higher level of bacteriocin production, as a natural selection. And then you can be sure that, in fact, you're sure because it's a primary metabolite. Will be always expressed if the bacteria is growing in its living. In case you want to stimulate, this will be again natural process because if the presence of the pathogen is around, bacteria will recognize the presence of these surface proteins and will stimulate its own production. The best way is select the best bacteriocin producer. You selected your bacteriocin producer, made it a spread in the plate, take several colonies, check which one of them is the best one, and continue working with the best one. Natural selection is always the easiest and more comfortable way to select the good producers.
We have the request.
Okay, good afternoon, participants. We are having our guest, Professor Narasimhan B with us. I invite him on the dais. So after the lecture, we are going to please. Professor Narasimhan B is the director IPSC of our university and also the local coordinator of Gian program we applied together. He was also key encouragers and apply for the different Gian program throughout the university. So we completed a 9 Gian program. This one is 8th already completed on the different topics. And thank you very much for all the assistance you provide us. During this program, we completed 12 lectures and 8 tutorial sessions. On the 13th, there was a lecture on systematics of lactic acid bacteria and their differentiation. On second day, it was isolation identification of probiotic lactic acid bacteria. And on third day, it was application of bacteriocins from food to the human health. And fourth day, it was probiotic concepts and the bacteriocins, which are essential for the research point in effective probiotic and role of bacteriocin in different aspects. On the fifth day, the several applications of bacteriocins from the beneficial to the clinical aspects from the human health, all they were covered. During the practical sessions, participants were taken to the laboratory and they understood how these probiotic bacteria can be grown and what are the metabolites that these probiotic bacteria can secrete and that can be used for the industrial applications. So out of the 12 lectures and 8 tutorial sessions, this was the requirement of Gian program. This 1 week Gian program covers this 12 lectures and 8 tutorials. The students were able to understand not only the theoretical aspect of the concept, but also they try to learn some of the technique out of these concepts. So this one is divided into 2 parts. One is we are going to take the feedback of the participants. If you are if you want to share some of your feedback, you are most welcome. One by one.
Good afternoon, everyone. I am Himanshi, Master Student from Department of Genetics, Maharishi Dayanand University, Rohtak. I would like to share my experience of Gian course on bacteriocin of probiotic bacteria from food safety to human and veterinary health promotion. This 1 week course was very helpful and insightful for me. I got to learn about various things which will help me in future studies. I, I would like to express my gratitude to Professor Todoro for coming to our university to teach us in this workshop. Overall session was very amazing and informative. I am very grateful to our Ministry of Education, our university, and our Department of Genetics for giving us this opportunity to learn on this topic. Thank you, sir.
Anyone next? Anyone? Yes. See, you can.
Good afternoon, respected faculty members, research scholars, and all the NOs present here. This is Priyanka Elat. I have done my master's in medical biotechnology. Firstly, I would like to thank Department of Genetics, Maharishi Dayanand University, Rohtak, Ministry of Education, Government of India, for organizing such a knowledgeful Gian course on bacteriocins of probiotic bacteria from food safety to human and veterinary health promotion. I would like to extend my sincere gratitude to Santosh Kumar Tiwari sir and Professor S.D. Todorov for their unwavering support. All the sessions were thoughtfully curated, offering a good balance between theoretical and practical applications. At last, I would like to thank organizers and speakers for putting together such an valuable and intellectual event. Thank you.
Thank you. Yes, no.
Greetings to everyone. Thank you so much, sir, for giving us the opportunity to interact with Professor Slavi. Can we call you Professor Slavi? And sir, from the beginning till the end, from basics to applications, even the basics were so interesting because you never know what point we can get clicked. For example, for anatomy, I was not knowing that molten agar, using the molten agar, I could actually help grow lactobacillus for other objectives as well, not just for the production of bacteriocins, to clearing up the concepts, very basic concepts. Sometimes we study and we forget what are the basics. They should be clear. So thank you so much, sir, and always being interactive with us. And none of us, I'm sure none of us was bored in any of the moment throughout the lectures. Thank you so much, sir.
Thank you. Sir, some are advanced level research scholars. They have completed almost of the work. They are also learning. And some are very UG students. They have just joined us 6 months before. And they said that it is helpful to understand basic technique. So the course for Professor Todorov was difficult from the UG to the research scholars to convince them at the same time. But thank you, Professor Todorov, you have done. Yes.
Good
Good afternoon, everyone. My name is Aarti. I'm from MS. I'm a student of Masters in Genetics. I would like to express my sincere appreciation to the Ministry of Education and Gian program for this remarkable initiative. Uh, Learning from renowned expert Professor Todorov sir enhanced our knowledge and inspire us to pursue excellence. And we enjoy his lecture and we learn a lot. And now we can think about more and learn more about world of science. And this program uh, enhance our knowledge so that we can improve ourselves in academic as well as research field. And thank you so much for this opportunity.
Thank you. Is anyone else? Backside.
A very good afternoon to the venerated faculty members present here. Myself, Navya, a student of BSc Genetics second year. First of all, I would like to thank the Government of India and our Honorable HOD sir who has organized this program in our university. And I would like to thank the Honorable Speaker who maintained their clarity, patience throughout the workshop to give us knowledge. So this workshop was a perfect balance between the theory and practical sessions. Even I was not able to understand at that level because I'm just a BSc student. But the practical session was that much clear that even a single spot that I can't lack at that session. And theory was a little bit some high level, but practical was very good. And I would like to thank you all. Thank you, sir.
Thank you. If you understood practical, one day you will understand the theory. That will ignite better to read it more, go through. Anyone else? We'll take one or two more.
Good evening, everyone. My name is Megha Sharma, and currently I am pursuing my master degree from Department of Genetics in MD Rohtak. So when I got to know that foreign faculty are going to teach us, I was very curious, and that's why I got registered myself in this Gian course, which was conducted by Professor L.G. Todorov. As a student with very limited knowledge, or I can say very less knowledge about bacteriocin, it was very informative and eye-opening session for me. You clarify all the concept, even the complex topics also in such a easy way for that help us to understand real world relevance also. And you even clarify all my doubt, even the basic ones also. So I'm very grateful to you for this opportunity and to learn help us to learn from passionate and such an experienced researcher. Thank you so much.
Thank you.
Good afternoon, sir. I'm PhD scholar and then Dr. Santosh Kumar Tiwari, sir. Sir, firstly, it was a privilege to be here with us. Sir, basically, this Gian course is much and more relevant to my research plan. And what things I get from this workshop is like number of experiment of including mode of action. I gonna include in my study to sharp my research like simple experiments including galactose 1. And you also like extended the things on safety parameters, which is I think the important things include beyond these like applications and characterization part. So I gonna include all these things in my research to validate my work more. So, and thank you so much, sir.
Thank you. Last one. Anyone? All right. If there is no one,
Say fast.
Good afternoon, sir. So we are very much honored to have you here, and I was very much
Thankful to him. Right, sit down.
I'm very much thankful to be here with you. And on behalf of all the team, I would say thank you so much for delivering so much interesting lectures with lots of humor in it. And I enjoy your every lecture and the practical session too. And we are grateful, enriched with the learning experience, and looking forward for future in future also. Thank you, sir.
Thank you. After the lecture sessions, they took the laboratory, and the students were more interested to interact with Professor Todoro. He's a very good human being. He will go to the student and teach them each and every point of the basic what is required actually for the students. That was the great thing that he went to the student and all the training and the experiment, the instrument and the technique. He was sitting on the laminar and showing that how to perform. So this is the great thing these students will learn and they will remember this thing even in the future that someone came from the abroad and taught me or showed me this technique so I learned it. So this is a great that you not forget this thing in the future. And they were keen to be interacted with Professor Todoro. And one of the students today sir took the Haryanavi food, dal bati, dal and churma. She cooked at home. See how interested is. You can tell your name.
It's a very nice.
Mansi.
Master students.
Yes, master student subjective department. And she was interested to show that yes, someone is coming. So that one student came with the sweet. What is that? Buttermilk. Who take Mansi? Yes.
Yes, I was the one who took that.
No, एक रसमलाई था. Okay, see, so she took that rasmalai. So it's a across the border test. The students were interested to share with him and interacting not only on the scientific content but also on different social aspects as well. They were sharing. Now I would request Professor Todoro to share his view in the brief. So I will invite in the last Professor.
Well, everything that started is to finish, but actually, if I have opportunity to stay a little bit more, I will. Santosh, can I stay longer? You say yes, I staying. No, unfortunately, I need to go for the next trip on Sunday. I need to go to Japan. But that was a very nice experience, very positive. Very nice to talk with you, to chat, to even talking about other things, not only about the microbiology. But this is actually the idea. Charles de Gaulle, one of the biggest French presidents, he was saying, put the students together from different countries, there will be the wars, because they will get friends and they will not ever fight against each other. So, but this is the point, interaction with other students is always a good point. And in Europe, we call students colleagues because it's not only one way teaching. Professor teaches the students, but professor learns from the students as well. And this interaction is very important. So thank you very much for the nice hospitality. It was really a pleasure. I hope that this agreement with our university will work and maybe we can have more interaction in the future. And maybe they don't listen. Come to do the Sao Paulo some work. You're more than welcome to come there. Because Santosh not will allow you. He don't want to lose the good students. He wants to work for them. But if you want to do some work with us, could be collaboration. Don't need to be just move there. Could be sandwich program, these hotel programs. Even you can thinking about maybe possibility of even double degrees. So why not? It's allowed by the law. It's a very well accepted program. So let's start and collaborate more and more. Thank you very much.
Thank you very much, Professor Todoro. It's a great pleasure that if you want to stay at our university will extend the hospitality and all the invitation. That is not the issue with us. But you're very busy scientist, I understand. You have your future assignments to visit the places. So he is leaving us day after tomorrow. So tomorrow he is with us, will be there. But it was somewhat free, and we'll have some sightseeing and other plans he has already done. So it's less assignment for tomorrow he has. So we had feedback from the students, sir, and also from the foreign faculty. And I was happy to complete this program on the specific thrust area. This is a very new thrust area, and it is a burning. And in the future also it has several applications. And a lot of students can join together to solve one problem, because there are so many opportunity in the one molecule can be this the different aspects can be worked on a different aspects, and that can be applied. So there is no other bacteriocins available throughout the world which are clinically approved and functional. All are in the pipelines, so others can work together. There is only one niacin which is approved, but food safety purpose better than the clinical drugs. But this one can, the work can be done. One bacteriocin niacin discovered 100 years back, but still they are working to optimize the conditions and know its potential possibility. So, without taking much time, Professor Narasimhan is having a meeting just after this. So, I would invite Professor Narasimhan D, Director IKC and local coordinator of our Gian program, to suggest his view about the course. Thank you. Very good afternoon to all of you. So, regarding the program, I think so we all will have suggested from your feedback. I think that first of all, I like to thank Professor Todorov for sparing his time and yes, having a very good interactive session with you. And like just sir was saying that Tiwari sir, that even to the laminar flow on your teaching, right? Yes, practically as explaining, this is a thing that is in skill that we have to develop in our university also, right? So teachers are that when to be a supervisors or you can, so whenever there is a need, you have to show practically you have to explain. So that we have did, that is a great part that we have to learn from most of our university culture also that has to be implemented. We are doing it, but it should be more interactive, right? And also should be technically. And thank you, Professor Todorov, for giving your valuable inputs to our students. So definitely they will achieve a new heights in their career. Then also regarding second, I thank Professor Tiwari for organizing such a nice program. And even though even being a local coordinator, yes, I came for the inauguration, then I came for conclusion. But rest of the work he has taken on is because I need no need to worry. He is a very sincere worker. So give a big round of applause to both Todorov and Tiwari sir for successful completion of the program. And another one thing that I just compliment you because you have even during your holidays you are participant in the program. Even the Todorov sir also he was saying that today yesterday that during even the during what you can say the holidays the students are coming so with more interest and they are very sincere. So that is a very good compliment from him for you. So thank you all for making this program very successful. And that's all I want to say. That is best wishes. Thank you. So in the last, the big thank for you that you joined and you came, participated with the so active understanding and all those encouragements. एक खाली अपने लिए बजाओ. Second, this program was possible because of your participation. Professor Todorov alone cannot speak, if someone is not there, because somehow it came into a holiday as he suggested us. But you came and interacted, participated in well. That is the only success of our program is. But if anyone take the message of learning any technique, understanding this concept, and doing something that will encourage your future understanding of this molecule or this concept better. So with all this, I am very thankful to the university organizations. They supported us, the hospitality and all technical supports from the university staff we got from the PRO and this institution, so the admission institutions, they provided us this seminar hall and also all the technical supports from there. That Director at USC's office was always ready for us to support, to apply and to show the area where one can go ahead, encouraging different faculty members to participate. And in the last also with the foreign faculty, but because he accepted our consent that yes, we can come and show this, but demonstrate this workshop. That is big because the country he is coming is somehow, sir, it take 36 hours one flight from Brazil to Sao Paulo to New Delhi. So 36 hours means it's like the third day you are reaching by flight, and it is not easy for all to decide that going to unknown place and take 36 hours. So it's his acceptance of our invitation is great for the university. University has extended its welcome and also understanding some of the memorandum of agreement between both the University Sao Paulo and Marcida and the University Rohtak, so that the students can get benefit better as compared to only laboratory to laboratory collaboration. So with all these words, and thank you to all of you who are directly or indirectly involved in this program. Thank you very much. So एक और मैसेज था, कल एक दिन आपको एग्जाम है, उसके बाद you will be free. I voluntarily wish you all happy Diwali. Happy Diwali to all of you. And what I wanted to say that we will have 1 hour only this examination, but please reach to 9:30 so that we will strictly start at 10 o'clock and complete 11. So it is very good for you also to find it. Thank you very much.
Não,